Hello everyone, welcome to our next lecture. So today we'll be looking at cell communications, and our objectives today are learn about cell surface and intracellular receptors, our G-protein coupled receptors, and our receptor kinases. So how do cells communicate? All cells process information from their environment. Communication required for coordination activities is required for coordination of activities. Communication is most often via chemical signals that bind to specific receptors, like we've talked about before. A specific hormone will bind to a specific hormone receptor. Same with a neuroreceptor. So one you'll see often is acetylcholine, binding to acetylcholine receptor, for example. We have carbon dioxide and our hydrogen ions. These signals can come from outside the organism or from neighboring cells, short distances and long distances. So examples of molecules that can act as signals. So plants, we use ethylene. So example, this occurs in the apple. And in animals, we have epinephrine, which you've heard of in fight or flight response. So receptor activation and receptor types. So a receptor. So simply put, a receptor is a protein that receives and interprets information carried by signaling molecule, which is a ligand. So for example, we have a receptor in immunology called CD40, and this binds to CD40 ligand. So the ligand, we can see here, is carried by the signaling molecule. So the ligand is binding to the CD40 receptor. The ligand binds to the ligand binding site on the receptor, and this binding causes conformational shape change in the receptor, and again in the entire receptor. This shape change activates the receptor, and these receptors can be found inside the cell, so these are intracellular receptors, and on the cell surface, which are cell surface receptors. So intracellular receptors. So again, these are found inside the cell. So this is when the receptor and ligand, if it's a steroid, equals the steroid receptor complex. And so these receptors can be either in the cytoplasm or the nucleus. So here we can see we have intracellular receptor, small nonpolar molecule, able to go through the cell membrane into the intracellular receptor, activated receptor signal complex. Then we have the small nonpolar signaling molecules can freely pass through the plasma membrane and activate the cytoplasmic receptors. So these intracellular receptors use nonpolar signaling, or can use nonpolar signaling molecules. So these are small and pass freely through the plasma membrane. So again, this passes through our phospholipid bilayer. Again, these are steroids. So steroids are lipid soluble, so they can go through. Steroids are hydrophobic and can pass through the plasma membrane. These activated steroid receptor complexes act as transcriptional regulators. So essentially, these steroids, so again, we have a steroid, let's say epinephrine, a hormone or a steroid. So for example, testosterone binding to an intracellular receptor. This then goes into the nucleus and activates transcription, usually resulting in some sort of effect and change in the cell. And again, this controls gene expression. So general structure of some receptors. So we have the ligand binding site. We have the extracellular domain. So here's our ligand binding site. Extracellular domain. So here we have our cell membrane. So binding site. So this is where it would bind. Our extracellular domain. 3 is our transmembrane domain. So this is when we get the part that's crossing the membrane. And then we have our cytoplasmic domain, which is this tail essentially in the cytoplasm. So we can also use polar signaling molecules. So these are small polar proteins which cannot cross the plasma membrane. There are different types of cell surface receptors that are polar. So here we can see polar signaling molecules cannot cross the plasma membrane and rely on cell surface receptors. So again, cell surface use polar, and then like we said, intracellular use nonpolar. So again, these are the different types of the cell surface receptors, just to clarify. So there are thousands of different types of receptor proteins on the surface of any given cell. So a bunch of cells will have a ton of receptors for a bunch of different things, hormones, immune system markers, etc. Most can be placed into one of 3 groups of receptors according to the way they are activated. So these are based on their functions. So we have our G-protein coupled receptor, which sometimes we'll see as GPCR. We have our receptor kinases, which we talked about in phosphorylation, and our ion channels. So during activation of receptors, we have phases. So here we have signal transduction, the response, and the termination. And this also can be seen here as receptor activation, transduction, response, termination. So signal transduction and sometimes amplification. The signal transduction or amplification response and termination are the steps that occur after a signal molecule binds to the receptor. Although the ligands are different, the subsequent steps are similar. So like we said, we have our activation, usually some sort of shape change, response, and then termination. So G-protein coupled receptors, like I said, GPCRs. So these are transmembrane proteins with this general structure. So first, they have a ligand binding site, which is extracellular. We have a transmembrane region, which involves 7 alpha helices. So here we can see the transmembrane region with our 7 alpha helices. Then we have G-protein binding site in the cytoplasm. You can see here. So there's type of signal molecule used by G-protein coupled receptors include small molecules, many hormones. You'll see this a lot in physiology with certain hormones binding to G-protein coupled receptors, as well as neurotransmitters. Like I said, commonly we see acetylcholine and other stuff like that. So G-protein coupled receptors are responsible for the senses of sight, smell, and taste. So they work on our nervous system as well for that. So when a ligand binds to the G-protein coupled receptor, it is activated. So when it is active, it binds to the G-protein, which is within the cytoplasm. So that's important. These G-proteins can be bound to either GDP or GTP. So these are the guanine nucleotides. So this is guanine diphosphate and this is guanine triphosphate, similar to ADP and ATP. So again, a G-protein plus GTP equals active. Think of GTP as the energy, essentially, like ATP, and GDP is inactive. So this ligand binds, then the receptor then binds to the G-protein. This GDP is replaced with GTP, so now it's providing that energy, and this G-protein becomes activated, and the signal is then transmitted. This active G-protein then activates other proteins as part of a signaling pathway, which we'll talk about in a second. So some G-proteins are composed of 3 subunits. So here we have our alpha, beta, and gamma. And here's that GDP we initially talked about. And as we said, it gets replaced. So again, we had our ligand on the extracellular domain. We had our alpha helices in the transmembrane and our G-protein area in the cytosol. So again, alpha, beta, gamma. So the alpha subunit binds either to GDP or GDP. So we can see here it's GDP inactive. So this would be inactive. And this would be active. So the alpha subunit and the GDP, there are 3 subunits bound, which is in an inactive state. So remember, GDP is inactive. GDP is inactive. So once this receptor activated, GDP on an alpha subunit is replaced by GTP. And these 3 separate subunits become active, like we see here. This activated alpha subunit binds to and activates the target protein response. So here's our active target protein. So we can see it moving. Alpha removes from beta and gamma and initiates a response with the active target protein. So one example of G-protein activation, amplification, is the adrenaline signaling in the heart muscle. So essentially kind of like a fight or flight response to increase our heart rate. So the target protein in this scenario is adenylyl cyclase. So here we can see activated adenylyl cyclase. This activated adenylyl cyclase converts ATP to cyclic AMP, which is our CAMP, which is considered a second messenger. This cyclic AMP binds to protein kinase A. This activated protein kinase A then phosphorylates. So remember, kinases tend to phosphorylate the heart proteins, and this causes the heart rate to increase. And again, as long as adrenaline is bound to this receptor, the heart rate will remain high. That's why we have other hormones like noradrenaline in our parasympathetic nervous system, but that's more physiology. So here, again, we have our receptor. We have a bound activated receptor. Alpha is removed from the beta and gamma subunit when it's bound to GTP, when it's an active G protein. This active G protein binds to the target protein, and in this case, it converts ATP to cyclic AMP, and this cyclic AMP with protein kinase A, we can see here, becomes active, and it causes the heart rate to increase. And again, as long as this is bound, it will keep heart rate up. So we have amplification of this adrenaline signal as well. So a little adrenaline goes a long way. So this amplification occurs at several phases. Here we can see at 1, 2, and 3, where a small amount of signal causes a large response. So the cell response depends on the type and the proteins in it. So G-protein coupled receptors typically activate downstream enzymes or open ion channels. Their effects are rapid and short-lived and reversible. So in this case of amplification, each activated receptor activates multiple G proteins, which in turn activate adenyl cyclase enzymes. So each adenyl cyclase enzyme produces large amounts of the second messenger cyclic AMP, which activates many molecules of protein kinase A. And as we said, so here we can see a bunch of them binding. Now we have our activated protein kinase A with cyclic AMP. Then each protein kinase A enzyme phosphorylates and activates the muscle protein targets. So here we can see this one activation releases several molecules, which binds to several protein kinase A, then works with several cyclic AMPs, and causes amplification, and binds to many different protein targets, as there are many receptors on these proteins, sorry. So the termination of a G-protein signal. So the amount of time a signaling molecule remains bound to its receptor depends on how tightly the receptor holds its binding affinity. So this is that strength of that binding for the signal molecule. So most ligands do not bind permanently to the receptors. The signal turns off once the ligand is unbound. So remember, the ligand binds to the receptor. Other parts of the pathway must also turn off, and this includes the phosphatases and the phosphodiesterases. So in this example, in the heart, the signal molecule adrenaline detaches from the receptor after a certain amount of time, inactivating the receptor so that it can no longer bind to active G protein. Within a very short time, an activated G protein deactivates itself by converting GTP back to GDP. So as we talked about, we have our activated GDP. So again, when it converts back, we have the release of that phosphate. When GDP goes back, it inactivates it, and then it stops the cycle. Enzyme in the cytosol specifically degrades cyclic AMP, which stop phosphorylation, inactivation of the target proteins, protein kinase A. These phosphatases remove phosphate groups from proteins, causing them to become inactive. So here we see they come in, these phosphatases remove the attachment, and now it is inactive. So receptor kinases. So a kinase is an enzyme that adds a phosphate group to another molecule, i.e. causing phosphorylation. Phosphate groups come from ATP. So as we see, an ATP is being removed, a phosphate is being removed in this reaction where phosphorylation, and the resultant is ATP, where phosphates are now attached. And a phosphatase is the opposite, where it removes the phosphate and releases an inorganic phosphate, and this creates an inactive form. This is called dephosphorylation. So when a protein phosphorylated by a kinase, that protein becomes active. There usually results in a shape change or provides a new site for another protein to bind. So it causes a little shape change, and then another protein can now fit in. We also have phosphatases, which have the opposite effect and remove the phosphate, causing dephosphorylation. These are important reactions that we saw in glycolysis as well. So receptor kinase activation and transduction. So here's a picture of a receptor kinase. So we have an extracellular portion, which binds to the signaling molecule. We have the intracellular portion, which is the kinase. There's our kinase. And we have dimerization. So dimerization activates cytoplasmic kinase domains, causing them to phosphorylate each other at multiple sites on their cytoplasmic tails. So essentially it's the interaction when these merge together. So we see here they come together. We also see this in our toll-like receptor signaling and several other types of pathways. But if you go back to our immunology lecture and look at the TLR pathways, we'll see that those dimerize as well. Phosphorylated areas provide provide sites for other proteins to bind and become active. So once they dimerize, they're now ready. These ATP releases its phosphate, now it's ATP, is now phosphorylated with these phosphate groups attached to the protein. Now, once that happens, it allows other proteins to bind, so these cytoplasmic signaling proteins are now able to bind due to the shape changes. So where is a receptor kinase signaling used? So it's used in many different things, but it's used in the formation and elongation of limb buds that become our arms and legs. It's also involved in wound healing. So when we cut ourselves, platelet-derived growth factor, or PDGF, is released from platelets into in the blood, which signals the cells at the wound site. This causes triggering of cell division to repair the damage. So in general, receptor kinases initiate long-term responses, as opposed to G-protein, which are short-lived. Activation of proteins involved in changes. They're involved in activation of proteins involved in changes in expression. They're involved in cell growth, division, and differentiation, and shape change. So one example of a kinase is the MAP kinase pathway. So this is mitogen activated protein kinase, which you've seen. I've talked about it in our immunology lecture, but MAPK. So here we can see the MAP kinase pathway. So in this example, we're doing like kind of a cut. So we have our paper cut. Our platelets release platelet-derived growth factor, or PDGF. This platelet-derived growth factor binds to receptor kinases on the cell surface. We have dimerization again. So remember here, this is when we have our dimerization where these come together. And this then we have the ATP. In this case, it's GDP going to. In this case, and then we see phosphorylation, and then activation of other receptors. Then we have something in this scenario where the RAS protein is activated in the cytoplasm when GTP is bound. So when RAS and GDP is attached, it's inactive. RAS and GDP is active. Kinase series is triggered, and these kinases enter the nucleus, and this causes the transcription regulators for cell division to be activated. And this pathway becomes inactive once GDP has bound to RAS is converted back to GTP, just like the other pathway. So here we can see during a cut, and a small amount of signal received by receptor kinase is amplified when the signal is passed from kinase to kinase, and each is phosphorylated. So we can see here kinase 2, kinase 1, kinase 3. So again, the signal is passed by kinase to kinase, amplifying the signal. Then finally, we have our changes in gene expression. So a receptor kinase, for example, called KIT, is responsible for the production of the pigment of our skin. So mutations in KIT receptor kinase cause patterns of incomplete pigmentations of feathers, scales, and hair. So this is essentially responsible for all these different variations in color. So now moving on, ion channels. So here we have a picture of a ligand-gated ion channel. So these are receptors that alter the flow of ions across plasma membranes. So a lot of them, one you'll see is the hydrogen ions or sodium and potassium. These are very common. So basically when it binds, there's a conformational shape change which opens the channel. This allows the flows of ions in or out, and the channel remains open as long as the signaling molecule is bound. So again, ions move through, usually causing response, for example, an action potential in the nervous system. So the integration of signaling pathways. So signaling pathways do not operate independently. Signaling is very complex. In one organism, many different signaling molecules exist, each with their own receptors. Cellular responses to a signaling molecules can vary from cell type to cell type, so every cell can be different. The effects of these binding depend on the various proteins present in different cells. So one molecule can bind to two different cell types and have completely different responses, even though it's the same receptor and the same molecule. So for example, cytokines, this could bind to one cell. And for example, ILA can have angiogenic effects. It could also act as a chemoattractant on another cell, even though it's the same receptor. So this is just my crappy version. Same receptor, same molecule, different effect. And different signaling molecules can bind to a single cell and activate several signaling pathways simultaneously. So the final response of a cell depends on the pathways intersect with one another. And even we'll see in a neuroscience course, they can work in a summation way. So there's multiple receptors and they kind of have a balance. So if one receptor is bound more than another, based on two different molecules, they kind of, whatever one wins gets the response. So it's quite interesting. So yeah, that's the end of our cell communication lecture. I hope you enjoyed it. See you next one.